The human cell is an amazing structure which is busy carrying out various life-sustaining processes, one of which is protein synthesis. For this process, it is essential that amino acids which lie scattered in the cytoplasm are carried to the messenger RNA or the mRNA template, the site of protein synthesis. Till the 1950s, scientists did not know enough about how proteins were carried to the mRNA. The breakthrough came in 1955 when Francis Crick, who had earlier revealed the DNA structure and its properties, postulated the adapter hypothesis theory, in which he stated that it is therefore a natural hypothesis that the amino acid is carried to the template by an adapter molecule, and that the adapter is the part which actually fits onto the RNA. In its simplest form, this hypothesis would require 20 adapters, one for each amino acid. Soon after Crick published his hypothesis, scientists across the world worked to find this mysterious adapter molecule that adapts itself to carrying amino acid and reading it to the codon of mRNA. Success, however, came to Robert Hawley when in 1965 he discovered transfer RNA or tRNA, the adapter molecule, and also worked out its entire chemical structure. Today we know that tRNA, the smallest of all RNA molecules, is made up of about 75 nucleotides. Synthesized from a DNA molecule in a process similar to the synthesis of mRNA, tRNA is soluble in 1 molar NaCl and is hence known as soluble RNA or sRNA. Each tRNA has G at its 5' end and CCA at its 3' end. Interestingly, tRNA takes the shape of a clover leaf when examined as a two-dimensional structure, whereas it looks like an inverted L-shaped molecule in a three-dimensional structure. Regardless of its dimensions, a tRNA consists of 4 arms or sites: ribosome recognition site, anticodon site, enzyme recognition site, and amino acid binding site or carrier end. Each of the tRNA regions perform a particular function. Take the case of the ribosome recognition site, which recognizes specific ribosomes attached to the mRNA and brings amino acids to the ribosome for protein synthesis. On the other hand, the anticodon site consists of 3 nitrogen bases called the anticodon. During protein synthesis, the anticodon pairs with the codon of the mRNA strand. However, the anticodon of tRNA is complementary to the codon of the mRNA strand. Apart from the anticodon site, a tRNA molecule also consists of an enzyme recognition site, also known as the D-arm or dihydrouracil loop. This particular site is the recognition site for amino acid synthetase enzyme, which helps the amino acid bind to the tRNA. Incidentally, this binding takes place at the 3' end of the amino acid binding site. Did you know that there is a specific tRNA for each of the 20 amino acids found in the human body? Also, there is a specific tRNA called initiator tRNA, which initiates the process of protein synthesis. However, there are no tRNAs for stop codons. tRNA is thus an important molecule, and it plays a vital role in protein synthesis by bringing amino acids to the mRNA template in the cytoplasm.